Hello, I'm Michael Breakspear. I'm at the University of Newcastle in Australia, and I'd like to acknowledge the Awabakal people on whose country I'm working and talking today. I'd also like to thank the organisers of OHBM 2022 and the organisers of this educational course on time-varying connectivity in resting state fMRI. I'll take one particular approach to that topic and speak to you about models of dynamic functional connectivity, also could be called biophysical models or generative models. As way of introduction, I'd like to show this video of a colleague and a friend of mine, Roland Girk. This is sped up, and this is actually Roland watching a short film, but I like to show it to demonstrate how even at rest or when viewing stimuli, humans are intrinsically dynamic and fluctuating. We can see changes in affect, we can see respiration, we can see changes in heart rate with the flushing and unflushing of Roland's cheeks. So we are really dynamic, fundamentally dynamic agents. And that's because we live in an extremely dynamic and unpredictable world. And to move with that world, we need to move intrinsically. The outline of my talk goes as following. I'll begin by talking about models of large complex systems. In fact, I'll be talking about models of the weather, since I think this is a really nice analogy, an example of where I think modeling in brain science could go. I'll then talk about using that same framework to make a large-scale brain model. As an example of dynamic functional connectivity, I'll then speak about metastable brain waves. And I'll conclude by quickly overviewing two simulation toolboxes that you can download and play around with some of the models that I'm talking about today, sorry. Okay, so why to model a large complex system, and how would we do that? Imagine we wake here in Glasgow, perhaps, look at our outside of our Glasgow hotel room and see this beautiful sunny morning. What we're doing is taking a snapshot of the weather, but we know, of course, the weather is like the brain. It's a dynamic system and it's subject to change. It's, say, 10 past 10 in the morning. We want to know, what's the weather like for the rest of the day? Is it a nice day? Should I take my surfboard to the beach? Not that there is one in Glasgow, but anyway. Or should I take an umbrella and get dressed up for some rain? In order to do this, as citizens, we can turn on the internet and we can look up the Weather Channel. And of course, we can get a weather forecast. What are weather forecasts? Where do they come from? They actually come from a number of supercomputer models of the weather. And this is a visualization of one of those models. This is showing temperature, wind, waves, and you can see down the bottom, as I change between the different modes, this is the waves, you can see the progression of the forecast from a nowcast through to a forecast into the future. This is called windy.com. And it's just a visualization overlay, but it's a really beautiful way of getting a notion of just how dynamic and beautiful the world's weather is. Okay, so this is the supercomputer model, and how does it work? These are extremely sophisticated models that are decades old, each decade getting more and more complex. At the heart of the models is the world's atmosphere broken down into a grid and actually into voxels, a little bit like the brain's voxels, with the surface and then boxes or cubes going higher and higher into the atmosphere. In each of these boxes are a number of different processes. It can be the ocean, can be the land. Now we have clouds in these models. Of course, we have humidity, temperature, wind, and other atmospheric processes. Now, these are in each little box describing some state of the atmosphere. The atmosphere is a compressible gas, and it obeys something called the Navier-Stokes equation. Obviously, the form of this equation is going to be a mystery unless you've done applied mathematics. It's actually a form of Newton's second law. So it's F equals ma, looking very sophisticated. It's formulated in a way that takes account to the swirling and sideways convection of fluids in the presence of a forcing term, which is the heat. These models take current observations and use these to predict the future. So these are the weather measurements that are going into the models. The models are solved, they're integrated forward, and they're used to predict the next set of weather observations, likely in an hour or 2 hours or a day or a week. Weather forecasting is on the timescale of hours to days to weeks, and climate forecasting is on the timescale of months to years to decades. The two are related, but you don't solve climate forecasts with this fast timescale. That's another issue. So that's how we get these models. We basically we have a set of models. They're used to forecast the weather. We have a bunch of weather stations on the ground. There's also many observations being taken in the atmosphere from weather balloons and such like. And these are then used to update the forecasts. So there's this iterative process between furnishing the models with observations, forecasting future observations, and then updating the models with what actually happens. And the mismatch between the forecasts and the observations is a prediction error. The generative models like this lie at the heart of models of many complex systems that are used in physics. Can be used to model financial systems, can be used to forecast earthquakes. The pandemic with the coronavirus was modeled in a not so dissimilar way as this. So, hey, now we're going to finally get over to neuroscience. You might have wondered where I was going here, but the fact is we're in the field I'm talking about, simply trying to do exactly in neuroscience what meteorologists have done for weather forecasting. So we're going to have an underlying model. It won't be derived from fluid, it will be derived from what we understand of neurophysiology, but the derivation will follow the same principles. And of course, it won't forecast weather observations, it'll forecast neurophysiological observations and behavior. So the model maps into observations. Our neural field model, as I said, will map into either EEG observations, MEG, fMRI, perhaps BOLD, and of course behavior. Just as we have this process of modeling, forecasting, updating, something called data assimilation, we can do the same thing with brain forecasting. This is particularly true of epilepsy, which I won't talk of today, where we have models, we make forecasts, we take measurements, and then we update those models. Now, on to making the models. How do we do this? Many of you may know that we have good models of the single neuron. In fact, the first simple model of a single neuron goes back 60 years to Hodgkin and Huxley. They developed a model from observations of the squid giant axon, and that actually won them the Nobel Prize for medicine, or maybe physiology. It's a set of differential equations. Once again, the particular form doesn't really matter. I'll speak a little bit more about these types of equations in a few moments. But these, instead of encompassing the motion of fluid at the single neuron, they incorporate the cell membrane potential with sodium, potassium, and other ions like calcium flowing, either leaking across the membrane or flowing through ion channels. The ion channels can be voltage gated. That's the form of this M and N. So they open and close according to the membrane potential, but they can also be ligand gated, and they occur in the synapse. So when you have a postsynaptic potential, a presynaptic release of neurotransmitters, you get these ligand gated channels opening in the dendrite, ions flow in, they're near the dendrite or near the synapse, they activate voltage-dependent ion channels, and that can trigger an action potential. And that's exactly what this set of equations describes. The first sort of closed set of physiological equations to describe the firing of a single neuron. And what's remarkable is it doesn't just capture the single action potential, but it can be used to capture different types of neuron firing. Here we've got regular spiking. We can also have an intrinsic burst and then spiking, and we can have continuous fast spiking. And I really recommend a book by Eugene Hesikovich, 2004, that didactically walks you through all of these behaviors in the full Hodgkin-Huxley model and in some very elegant reduced models as well. Now, we're obviously doing human brain mapping. We're looking at large-scale neurophysiological recordings. We don't really, in our field, target the action potential of individual neurons. Neurons are embedded in local circuits. Local circuits are embedded in small structures like the hippocampus. So you can have a single neuron, then a subfield of the hippocampus, then the hippocampal circuit, and they're then embedded in a large network of connectomics. So we've got this hierarchy of spatial scales. Individual spikes are too weak to influence behavior. Otherwise, we'd be jumping around with the spiking of billions of neurons. Our behavior would be very unpredictable. But behavior likely reflects the coordinated action of many, many millions of neurons. This brings down the influence of noise and increases the robustness of our coding of behavior to high-dimensional fluctuations. There's now a popular view in the field of computational neuroscience that says individual neurons play a very important role, but their role is much like one voter in an election. It's like a democracy of neurons, with each individual neuron contributing to some local collective decision. And of course, there can be, just as there are in political elections, there can be more than one party, two parties, three parties. In Australia, like many countries, it looks like we're going to have rule by a number of collaborative parties. And that can also be the case, we think, of neurons. I'm going to get to that. This population approach is conveniently matched to the apertures, the spatial scale of human brain mapping technologies, fMRI, EEG, and MEG. And I think some people might say, oh, people like Michael Breakspear just take this population approach because it's convenient for the type of measurements that they're doing. But I think there's quite compelling underlying theoretical arguments to take this approach. So how do we make one of these large-scale brain models? We take a local neural population. We're talking about hundreds or perhaps a few thousands of neurons. We can code up the Hodgkin-Huxley model, or we can make a few simplifications and we can code up something a little bit like it. And on a regular server, we can indeed simulate the spiking activity of hundreds or some thousands of neurons. This is called a spike raster. Each little black dot here is the spike of one neuron. So you see here there's 1,000 neurons, that's the y-axis. The x-axis is time. So you see here there's some sort of intrinsic oscillation, but each neuron itself is still quite noisy. And in this population, there's a few hundred inhibitory neurons, and there's, well, 800 excitatory neurons. So this is a small population. Now, if I take two cross sections at any particular time and take two little time windows, I see that the underlying neurons have a distribution of states. This could be the membrane potential. It could be the fraction of potassium channels that are open in each neuron. It could be some current. It doesn't really matter. I haven't put units here. This is just a probability distribution, a histogram. There's a histogram on the left here, shortly after we've started the simulation, and there's a histogram on the right a few hundreds of milliseconds later. Now, the magic comes in doing something called a mean field approximation. It's close to the central limit theorem. It doesn't assume that the individual neurons are all identical. It just assumes that to some extent, those properties come from an underlying distribution of neurons or properties, and their spikes aren't all completely dependent on each other. So there's some degrees of freedom, but there's not complete degrees of freedom. So instead of coding up 1,000 sets of equations, we're going to reduce this population to just the mean and the variance, or the value of those states, which is the mean or some other central tendency, and the variance or the precision, which is the inverse of the variance. And then we're going to try and find a set of equations that propagates that forward and predicts the mean and the variance of the actual histogram a few hundred milliseconds later. Just go back here again, walk this through. Here's the distribution of states. Here's a mean field approximation. Here's some reduced form of that population. And here's a fancy equation that predicts what that population will be. And if it matches what the full population is, then we've got a pretty good model. This is not particularly unknown. It's actually very common, widely used in physics, the behavior of magnets, superconductors, liquids screen, computer displays, ant queuing, swarming, all sorts of systems with many individual agents. Okay, I'll walk this through. Before I go any further, in many cases, we have to discard the variance and we're just modeling the mean. This is now what's called a neural mass equation. Okay, so let's go through this now. We've got our complex neural histology. We've now come up with a quite complicated mathematical model, not just one set of equations per neuron, but a set of equations for every little different compartment of neurons, for the dendrites, for the cell soma, for the axons. This is what's done in the Human Brain Project as it was originally conceived. Then we can reduce those. We can get a single set of equations for each neuron. There's software for doing this, Brian Neuron SpikeNet. And then we can do what I just showed, which is to get rid of the spiking neurons and have a smaller set of equations to describe the population density. There's several types of equations here, the Janssen-Ritt model, the Wilson-Cohen model, different approaches to doing this mean field approximation. These are following an approach called the mean field dimension reduction. And you might think, hey, you've thrown out all or much of the underlying neurobiology. What are you doing here? You're ending up with something that's so simple, it's not really brain-like at all. Well, when we go to these more complex models, we can, in fact, and do, capture the way dendrites filter presynaptic potentials, the way those currents in the dendrites get summed at the cell soma and then converted into outgoing action potentials. And we can certainly model the way they propagate across the surface of the cortex according to the likely length of axons. But we can actually do more. And in this recent review paper by Max Schein, you can read how neuromodulation at the cellular scale can be captured by these mean field approximations. We know, for example, one can up and down regulate postsynaptic potentials. EPSPs can go up and down according to second messenger systems. The action potential can be bumped up or down, the threshold, for example, by an influx of calcium. The refractory period can be shortened under the influence of certain neuromodulators, and the threshold can be repressed or moved down by other neuromodulators. These are neuromodulatory processes that arise through things like serotonin, dopamine, noradrenaline, other ascending neuromodulators that don't on their own cause action potentials, but change the gain and the firing properties of individual neurons. We can incorporate the way these neuromodulatory transmitters are distributed across the brain by incorporating, for example, PET maps. And we can then propagate these processes through the mean field reduction and capture them in our neural field and neural mass models by changing functions called the gain function. So all is not lost. And if you'd like to read more, please read this recent paper by Max Schein and colleagues. Okay, so we've got what's called the evolution equation. And what do we do with it next? This is an equation that captures the neurophysiology in a particular part of the cortex. Now we're going to just walk through some basic concepts in applied mathematics, because we understand how to use mathematics and not just go straight to computer simulations. Now, if we think of membrane potentials, ion channel conductances, these are the states of individual neurons, and their means are the states of the neural populations. They make something called a phase space. If we have two, then we have a plane. If we have three of these states, then we get a volume. These equations, for every point in this phase space, have an amplitude and a direction. In other words, they have a vector. So this set of equations defines something called a vector field. It's a flow. It's the way the system will flow through the states that it can possibly occupy. Now, coming from these vectors are orbits. These are solutions to these equations. They are loops and closed orbits that are defined everywhere by these vector fields. And if we understand enough of them, then we see that these orbits flow onto something called attractors. So we've gone from phase space through vectors to orbits to attractors. An attractor is a closed orbit that attracts a lot of surrounding orbits. It's quite an intuitive concept. The attractor then captures how the system will behave after some short period of time. If it's closed, we get a limit cycle. If it loops around like this, we may get something called spike and burst and spiking. If it's more complex, we get something called a chaotic attractor. So now we've got our neural mass equation, we've got our underlying attractors. Here's an example of one that I've played around with that you can read in some of our older papers. Here it's exhibiting burst and spiking behavior, like a 3 Hertz theta oscillation with a change in the underlying parameters. The same set of equations now bifurcates into something called a chaotic attractor. The shape of the attractor maps onto the type of time series. Now, just as we did with our weather system, we take this local population, we put them everywhere over the cortex, hundreds of them, or perhaps thousands of them. They're all evolving away with their local dynamics. And then we measure the connectivity between them. And I'm sure most of you are acquainted with the connectome. This is a set of wires or connections that link different regions of the cortex. So now we're going to link these underlying chaotic attractors, and we're going to see what happens to them. This is an example. Of course, there's many of these papers. Here's a paper by James Roberts a couple of years ago in Nature Communications. And in this set of models was observed large-scale brainwaves, rotating waves, something called breathers, and traveling waves across the cortex. So these are predictions from these networks of coupled models. Now, it's not perhaps surprising that traveling waves have been described in many different species using many different data modalities, voltage sensitive dyes, local field potentials, even in perception, in binocular rivalry, you can perceive waves. And this is an example of an underlying dynamic model of waves. I won't go too much into the detail, but these waves come out of the way local patches of neurons are connected to their neighbors. In fact, a few years ago, in sleeping electrocorticograms, Lyle Muller and colleagues described traveling waves in humans, and the wavelength and carrier frequency of these waves are similar to those that we described in this particular model. So we're definitely not where weather forecasting is, but I hope you can see a few of the initial steps that we're making in that direction. These waves fluctuate from one type of wave to another, from traveling waves. A few seconds later, there's breathers, then there's rotating waves. I won't go into detail because I'm running out of time, but we can actually use the vector field to understand those transitions. And here's an example. The waves on the left, the vector fields in the middle, and the orbits on the right. And you can see these beautiful dynamics being predicted by this model. And this is a type of spatiotemporal chaos. There's long periods of stability, then these underlying vector fields get tangled and you get a rapid transition to a different form. That's what we see in this particular model. We get these changes in states, which we can partition. And this is now a generative model for dynamic functional connectivity, if it was measured on the correct timescale. So that finishes my whistle-stop tour of dynamic models of time-resolved functional connectivity. If you're interested and want to go further, The virtual brain is extremely well developed, Python-based interface, which comes with the ability to do everything that I've discussed. It was developed as part of the European EU Brain Project by Vic Yersa, Randy McIntosh, Petra Ritter, and others. If you just want to play around with some simple models and you're still a MATLAB maven, then I can recommend the Brain Dynamics Toolbox, which I wrote together with Stuart Heitman. That can be downloaded for free. And I'll just quickly show how the Brain Dynamic Toolbox comes shipped. All of these different windows for understanding exactly what I've just shown. It comes with its own models, Wilson-Cohen model, other similar models. You can then integrate these models. You can understand the phase space, the time series, and you can also subscribe and receive updates and also a series of tutorials. And it's backwards compatible to MATLAB 2014. So here are these resources. Google will take you to each of them. This work was funded by the ARC Centre of Excellence for Integrative Brain Function, the NHMRC, and these are members of my group here in Australia. And I'd like to thank them for coming to Newcastle with me and setting up the group. So thank you very much. And I'll take questions.